Thank you, Dr. Chan, for the nice introduction. As she said, my name is Alec Tan, and my project this year was evaluating the efficacy of Oahu invasive species stream removal efforts using environmental DNA. So my inspiration for this research ultimately came down to two topics. The first was the fact that I spent last summer at a research internship at the UCSF Proctor Lab of Dewan, Dr. Dewan. And here I learned about all the details and processes behind metagenomic deep sequencing. And I became fascinated with this kind of technology, and I wanted to apply it in school, especially in a school research project. And my second interest came when I found out about native Hawaiian amphibious fish, which some of the fish are able to scale vertical waterfalls as a part of their life cycle, as shown on the image on the left. And I thought this was a pretty cool feature, and I wanted to learn more about them. So that's why I picked this topic. So Hawaii is home to 5 native amphibious fish. And amphibious fish are fish that have a marine larval phase, but migrate up through freshwater streams as a part of their juvenile and adult life phases. And some of them climb waterfalls to get up higher into the mountains. And it's a pretty unique feature. Now, these 5 fish are endemic to Hawaii, meaning they exist nowhere else in the world. And they vary in levels of threat status. Specifically, O'opu no pili and O'opu alamo'o are among the more threatened species. Ultimately, these species are threatened because of human activities and influences. One example is urbanization of Oahu, such as which has altered the natural landscape and watershed and made it harder for the O'opu to continue their migration upstream. And another, and probably the biggest factor, is the introduction of invasive species from the aquarium trade into the streams. This includes like catfish, guppies, and bass. And these fish have outcompeted the native fish and thus resulted in a rapid decline in their population. Now, this has led some community organizations to take action and directly remove the invasive species from the streams. So one organization known as Pai Pai o Vaikolu has worked to teach K-12 students about the importance of preserving the Hawaiian watershed and its biodiversity. And it teaches students how to operate nets, large fish nets. Then they go out into the streams and remove invasive species from the streams. And with their work, they've been able to remove over 40,000 fish from the invasive fish from the streams since 2015, which is a large number of invasive fish. And from their results, they have found that on average, they've captured more native fish in their surveys, and also they're finding less invasive fish in their surveys as well, meaning their work is likely improving the biodiversity of the watershed. Now, I should note that due to the pandemic, they were forced to cease their efforts for several years, and this has led to an increase of invasive fish, but they have recently renewed their efforts to remove invasive fish from the streams, and hopefully that will lead to some improvement. Now, one issue with their census method is the amount of manpower that it requires. Each group that they take out to the stream cleanups requires 3 one-hour sessions teaching them about the biodiversity and the watershed, and also how to operate the large nets, and also a 3-hour stream cleanup session where they go out into the streams. And you need adult chaperones and teachers to teach the volunteers how to operate the nets and also ensure safety. And this requires a large expense of manpower. So one might wonder whether or not there's a better, or not better, but less manpower heavy way of monitoring the streams. So one alternative survey method to this is environmental DNA. Now, environmental DNA is DNA that has been released into the environment off of an organism. So this includes like fish scales or fish excrements. And with this, with eDNA, we can sample the environment, extract the DNA from that sample, and then use it to identify what species are in a certain location. Now, environmental DNA has had some other uses, but in this case, we'll be using it for invasive and native species detection, particularly because environmental DNA only needs the slightest trace amount of DNA in order to detect an organism. So we can use it to detect some of the rarer native fish that we might not normally see in a stream survey. So this leads me to my research question. Can environmental DNA be used as an alternative method to compare traditional manual surveys of native and invasive fish species in the LOI watershed? Now the LOI watershed being the three Makiki, Manoa, and Pololo streams that lead near the LOI Canal. So for this project, I selected several sites circled blue, purple, and circled in blue and purple on the map where the traditional surveys are also conducted. At each of these sites, water samples were collected in order to extract them for environmental DNA. After collection, each of the samples were underwent consolidation methods through either push filtering and syringes, vacuum filtering, or centrifugation in order to consolidate the DNA from the water sample. Once the DNA has been consolidated, we then used a DNA extraction kit, and this the D&EZ blood and tissue kit to extract the DNA from the consolidated DNA from the sample to get the DNA just by itself. Now, since DNA is generally in low volume or low concentrations after it's been in a dilute stream, we want to create more copies of the DNA so that we can work with it and analyze it. So we put it through polymerase chain reaction or PCR to create more copies of the DNA. And in my experiment, we used myFish primers, which are general fish primers that target a region of the 12S rRNA gene that can be used to identify fish species. So this allows us to replicate more fish DNA and mostly none other organisms' DNA. And after we've confirmed that there's DNA in the sample, using verification methods like gel electrophoresis or quantification, we can then prepare it for sequencing, where the sample fragments are identified as the base pairs are identified. So we prepare it for sequencing by putting it through library prep, and then once all the samples have been put through library prep, we can consolidate the samples into one solution and run it through a sequencer. In this case, it was a Minion nanopore sequencer. And this sequencer contains many artificial flow cell channels where the DNA is run through these channels, that produces an electrical signal that is interpreted by the computer as one of the base pairs DNA nucleotides. So A, C, T, or G. And from that, we get the raw reads of each of the DNA fragments. Now, with these DNA fragments, we can filter them out using the Dacona pipeline process, where each of the DNA fragments we conduct quality control of all of our reads of the fragments and filtering them out. Then we can cluster similar fragments together into a consensus or average of the DNA fragments. And we can then use a locally curated genomic database containing fish genomes and to match it against our averages of DNA fragments to see if they align, and then we can identify each of the fragments as a different species. So my results initially found that environmental DNA detected more species in Manoa marketplace compared to traditional captured survey data. So environmental DNA was able to detect all the same fish that a capture survey that was conducted on the same day also detected. And in addition, it detected a few other fish species that eDNA, that the capture survey did not detect. Now, this was not always the case. In another two samples from Kaimuki High School and Kanawai Field, environmental DNA detected less species than the capture survey detected. In this case, environmental DNA was only able to detect 4 species, one of them being human, and the capture survey was able to detect 10 different fish species. So there's a significant drop in detection rate here. And additionally, we also found that eDNA may be subject to bias. In this graph here, the number of sequence fragment reads of each fish species at the Manoa marketplace is shown, and it shows a disproportionately large number of catfish and bushy plecostomus reads compared to the actual number of fish, which is like in the single digits in real life. So, and this kind of shows how these quantifiable numbers of species are can be biased, because there are factors such as PCR bias, where some species DNA are favored during the PCR or replication process than others, as well as the amount of genetic material each kind of fish can release into the environment. So catfish are more slimy than like a very tiny Mexican molly, so they're likely gonna release more DNA into the environment. So this kind of highlights the potential issues that you might encounter when working with environmental DNA. Environmental DNA is a time-consuming process because although it requires less manpower to initially collect the sample, it definitely takes a lot more time in the lab to complete each of the steps of the methodology that I listed before. And there's also many possible sources of error that can occur in the process, which is why we were unable to get results for many of the other samples from different sites that I've collected from. And this adds to the inconsistency of environmental DNA. So in conclusion, environmental DNA can be used in comparison to traditional surveys in some cases. However, as seen in the Manoa marketplace sample, however, environmental DNA can't be used alone due to its inconsistency shown in the other two samples from Kaimuki High School and Kanawai Field. So environmental DNA is best used as a supplement in addition to traditional surveys in order to validate the results. And some future research topics could include investigating the inconsistency between the samples and trying to develop a more polished protocol for conducting environmental DNA experiments. And we could also try to create a complete picture of the other streams in the LY watershed. For this presentation, I was only able to get data from the Manoa streams, although I did sample and extract and filter samples from Makiki and Palolo streams, but those samples did not come up with any DNA inside of them or in those samples. Therefore, I could investigate trying to get actual data from those other two streams. Now, some advice to future honors independent research students. My number one would be don't procrastinate. There are lots of deadlines in this class, and to keep on task with your project. So you should if you have time to work on your project, you should definitely do it. Another tip I would give is to pick a research topic that you enjoy and that others would find interesting too. Pick something you enjoy because you're going to be doing it all year, but you should also keep in mind that some other people need to find it interesting too, or they need to see the value of your research. And otherwise, why are you doing the research if no one cares? So, and finally, you should be willing to ask for help. There were plenty of times when I was in the lab and I forgot how to do something, or I forgot what step I was on of a process, and I was always willing to ask help from like Dr. Chan or Ms. Kobayashi, and they weren't afraid to help me either. So don't hesitate to ask for help. And finally, I'd like to give acknowledgements and for everyone who helped me along my research project journey. I'd like to thank Dr. Chan and Mr. Hill for all the help they've given in guiding my project, as well as Ms. Kobayashi. And I'd like to thank Mr. Yap and Ms. Sharuk for providing me with stream data and advice on where to conduct my samplings from. And finally, I'd like to thank my family and Honors Independent Research classmates and friends for their support along the way. That's it. Thank you. I'll open the floor for questions. Thank you.